All right, hello all, and today we are going to be continuing with the second part of the video. All right, now before I proceed, actually, let me remind you that actually we missed a few of the questions, so I am going to get back to those questions that we missed yesterday. Particularly, there was this a question in particular, so we missed the part the part C of this question. It was a rather very interesting question. Yeah, I forgot to include it in the worksheet, but now we're going to go for it. All right, so let me proceed. So small vacuoles in S. pectinata may have roles similar to lysosomes in animal cells. Describe the role of lysosomes in animal cells in defense against pathogens. All right, so it's very specific now. So you may forget all these parts. It's not really important. It's just a question about lysosomes. So describe the role or the function, what's the function of lysosomes in animal cells in defense, particularly in defense against pathogens. It's not a long one. It's just 2 marks. And lysosomes, they just contain hydrolytic enzymes. Okay, they just contain hydrolytic enzymes, about 60 plus hydrolytic enzymes that are stored in that single membrane bound organelle. And yeah, essentially that. And here is talking specifically about the defense against pathogens. So what happens essentially is just after the macrophage, for example, a macrophage has ingested or engulfed a bacteria, for example, or any other pathogen. So what will happen is that lysosome will come and fuse with this phagocytic vacuole, and hence release the enzymes, the hydrolytic enzymes, and thus causing digestion of the pathogen. All right. So I'm just going to go on and put an answer here. So here we go. Insert home. All right. So. Not a fan of that one, so here we go. All right, and I like the blue color a bit more. Not this one though, perhaps this. Okay, and then a bit of yellow to it. Okay, this is not bad really. And one more thing, sorry about that. Shape format, shape fill, more fill colors, also 50%. Okay, here we go. And oh, one more thing actually. Sorry about that. Just shape outlines, just no outline. The shape. All right, now we can proceed. So describe the role of lysosomes in animal cells. So the role is quite straightforward, isn't it? So it just helps in the digestion. How? The fact that it contains hydrolytic enzymes. So see. So lysosome are membrane bound organelle that membrane bound organelles that some reason my device is lagging a bit, that contain hydrolytic enzymes. Okay, full stop. After pathogen or subsequent, after pathogens have been, for example, engulfed by maybe phagocytes, phagocytes, or you may say macrophages. Then what happens is that the lysosomes fuse with them to release these, to release the hydrolytic enzymes. Full stop. Hence, these enzymes help in the, you may say digestion, or you may say destruction, helps in the destruction slash digestion, or you may say hydrolysis, hydrolysis of the pathogens. Okay, full stop. Now for 2 marks, well, I'm not sure what does the mark scheme say, but let us go on and have a look at that. Okay. Oh, let it go up again. I'm gonna have a look at that. Okay, here it is. Yeah, here it is. So it talks about the formation of phagolysosomes. Okay, you may say that, and you talk about the breakdown, digestion of pathogen, which I did. You may say kill or destroy, that's fine. And 2 named enzyme, no, I did not. You just need 2 mark, don't you? Yeah. Oh, I forgot to mention the phagocytic. All right. Okay. Lysozyme, protease. Harmful. Pardon. So harmful. It's harmless. Okay. I think. Okay. To form harmless products is interesting. Okay. I should have mentioned that. I think I'll get only 1 mark if I keep what I wrote. Although say here it says the part regarding formation of phagolysosomes is described. They would give the mark if it is described, but yeah, I'm a bit skeptical about that. Okay, so anyway, let us to keep track with the wordings of the mark scheme. I'm going to be using words here mentioned here. Okay, so phagolysosomes. So what are phagolysosomes? All right. So a phagolysosome is the sort of a cytoplasmic body that is formed by the fusion of a phagosome with a lysosome. Okay, and in a process that occurs during phagocytosis. Now, so what is it that happens during a phagocytosis? It is recording. We are recording, right? Okay. Give me a minute. I'm just gonna refer to the notes here when I get back. All right. Okay, so I'll read that out to you. So, phagocyte, phagocyte, they carry out phagocytosis. So essentially, once attached, so we've got receptor proteins, but we are not going to get into details about those ones. So they extend out around the pathogen, engulfing it and trapping the pathogen within a phagocytic vacuole. Okay, so you just may say that, yeah, neutrophil, they secrete digestive enzymes into the vacuole, and then this occurs when the phagocytic vacuole, also known as a phagosome, fuses with a lysosome. So the fused phagosome and lysosomes are referred to as phagolysosome. So I'm just going to right, so back here, so I'm going to put it as such. So during phagocytosis, phagocytosis, um, during phagocytosis, so the phagocytic vacuole, or you may say the phagosome, I'm going to use the word phagocytic vacuole, fuses with lysosome. During phagocytosis, the phagocytic vacuole fuses with a lysosome to form a phagolysosome. All right, this part is okay. To form fuses. Did I say fuses? Yeah, it fuses with a lysosome to form a phagolysosome. What did I say? It fuses with the lysosome. The fused phagosome and lysosome are referred to as phagolysosome. Okay, and then simply what happens is that the hydrolytic enzyme are released. The hydrolytic, you may use the word hydrolytic or enzyme. So fuses with the lysosome to form a phagolysosome. So full stop about there. Let me see, what does the mark scheme say again? So what does it say here? Where is it? A phagolysosome. Okay, formation of a phagolysosome, and then breakdown or digestion of the pathogen. Okay, full stop. The hydrolytic enzyme from the lysosome are thus released into the phago. They are not released then from the lysosome, thus causes the digestion of the pathogens, thereby or subsequently thereby not transform, how do you say it? Turning the pathogen into harmless projects. Okay, so yeah, just harmless. To form, right? To form is the word, right? Let me see what did they say here. To form harmless products. Okay, to form harmless products indeed. Okay, so then the hydrolytic enzyme from the lysosome thus causes digestion, thereby forming harmless digestion of pathogen. Let me just put it there in the same sentence. To form digestion of pathogen to form harmless products. Okay, and there you go. And that would be your answer. All right, now let us proceed without much of a delay. Okay, I also added this question. So now, here you may say, but this chapter we have not yet covered, so why is it here? But anyway, this is May June 20. Pardon, it is February March 2022, and the second variant of paper 2. It's talking about TB, tuberculosis, and it's a major cause of ill health worldwide. So it's supposed it should be a more of a general knowledge. Okay. So what is it that causes tuberculosis, TB? It's actually caused by a bacterium called Mycobacterium tuberculosis. Okay, Mycobacterium tuberculosis. So say the name of the bacterium that causes TB in humans. So I'm just gonna write the answer. It's quite straightforward. So what is Mycobacterium tuberculosis? There you go. All right, next. So whatever is a scanning electron micrograph of bacteria that cause TB. Okay, and what is it? Calculate the actual length. Okay, so we are not going to be able to do this, but what would be the formula that you'd use? What did it say? What's the question about? Calculate the actual length of the bacteria. So it's given the magnification here. So given that the magnification is equal to multiply 21,000, okay, and you're supposed to measure that distance xy, and give your answer in micrometers. So you should make sure to just ensure you convert everything into, yeah. So you know this formula MIA. So we are looking for A. So it's actually M is equal to I upon A. So actual length. So the actual length. How are we going to calculate it? Actual length is going to be equal to simply the magnification. Pardon about that. Magnification. That is you may just put 21,000 out. 21,000 divided by the thing that you measured. Okay, the measured, so the image size, you may say image length, which should be in micrometers. All right, so that is the formula that we'll be using. Next. Here it's an interesting question. Bacteria are unicellular prokaryotic cells with diameter of over 1 to 5 micrometers. By the way, this is a value you should quite be familiar with because it is included in your specifications. So you are expected to know about it. So state, to other. So one of this is the diameter. Two other structural features that would identify a cell as prokaryotic. So no nucleus and perhaps you may say no organelles. Okay, structural features. Yeah. It's not letting me move. I hate this. Okay, are you serious? Oh my god, are you serious? All right, so what are the two things I'd say? Perhaps the absence. Okay, the absence of a true nucleus, and secondly, you could say absence of organelles. But using the word organelles is a bit too broad. I'd rather say, for example, absence of well, they do have ribosomes. For example, you could say absence of rough endoplasmic reticulum or Golgi body because they don't have that Golgi body. And prokaryotic, especially if you talk about, speaking of bacteria, bacteria, they specifically have a cell wall of peptidoglycan. So, but I'm not sure if they accept it because it's just talking about prokaryotic cells in general. Well, yeah. Now let me quickly go and check the answer key for this question. Yeah, here we go. So Mycobacterium tuberculosis length. Okay. Any two from. Okay, they are accepting the peptidoglycan cell wall. Absence of named example of membrane-bound organelle. So I gave an example like, what did I say? Golgi body, right? Yeah. And accept no true nucleus. Okay, yeah. They're accepting it. Okay. So I'd rather refrain from using this because it's just like an additional accept, but it does not form part of the actual mark scheme, sort of. So I'd actually refrain from using the word and the true nucleus. Okay, just memorize these ones. Okay, cool. Now we are done. We are essentially done with those initial parts, and that'd be the part one of the worksheet. Now we can proceed to the second part of the worksheet that we were initially supposed to be doing today, but yeah, there was those left out questions that I had to complete before moving on to the next part. Okay, so now we are going to proceed with the part two. So here is the part two of the worksheet. Okay, now it is the 2023 paper of the October-November series, and it is the first variant of the paper two. All right. Proceeding, figure 2.1 is a transmission electron micrograph, a TEM, a transmission, okay, showing a section of a specialized epithelial cell found in the lining of the stomach, and this cell produces extracellular proteins that are released into the bloodstream. Oh, okay, well, that is interesting. Magnification. Okay, outline the role of structure X and structure Y are shown in the figure in the production of extracellular proteins. Okay, what is this picture again? A section. Okay, so it is a section of a cell, of an epithelial cell. So this is a cell. This cell, okay, this is a cell. All right, just thank you for confirming that. Now we can proceed. Structure Y and structure X. Well, that is quite a thing. Now, this whole structure out here, maybe this whole structure, which I'm trying to paint in yellow, is our trusty old nucleus. Okay. Okay, this structure is the nucleus, and yeah. Now this darker stained region, which is what X is referring to, and then there's this lighter stained region, which is referring to, which is what alphabet Y is referring to. All right. So this structure X is just the nucleoplasm. Nucleoplasm. No, it's not nucleoplasm. Pardon. Oh my God, what's the word? Nucleo. Nucleolus. Yeah. Oh my God, that was a blackout. A nucleo. What did I just say? Nucleolus. It's nucleolus. Okay, and this structure Y would it's quite really ambiguous here, but I'd perhaps go on and say it's just a chromatin. Well, even nucleolus is just a more dense area of chromatin fibers, but yeah. Oh my God, what did I write? Chromatin. Chromatin. There you go. All right. Great. So now what outline the role of structure X and Y in the production of extracellular proteins. Okay. I'm not sure of the answer, but yeah, so that which is why I'm going to be referring to the mark scheme. I did include all the answers, didn't I? All right. Oh, okay, it is indeed correct. Now we've got the two structures out here, which we figured out quite clearly. First is the nucleolus, and second is the chromatin. And the function of the nucleolus is simply to work as the synthesis of ribosomal units or subunits, pardon. Okay, it's just this function is simply for the synthesis of ribosomal units. Okay, and chromatin, chromatin is just essentially DNA, and it just contains genes. Okay, or you may say it codes for the polypeptides. It codes for polypeptides. It contains genes which are responsible for it contains genes that code for polypeptides. Okay, contain genes that codes for polypeptides. So firstly was which structure again? X. What's X? A nucleolus. Nucleolus, and it's just the site for the its role. Its role is simply for the. Did it say site for a synthesis of ribosomal subunits, ribosomal subunits. Okay, and next structure, of course, you write better. It's just a bit difficult for me to write properly here. And next structure is the chromatin. I don't have my graphic tablet yet, so for polypeptides. Okay, that's it. Next question. How do we reach there already? Okay, next question now. Since I've not put any new reference here for the date and stuff, so I suppose it's just the same paper. Anyway, plants have specialized cells for the efficient transport of assimilates, and table 3.1 shows some of the features of two different types of cell found in plant tissue, which are adapted for the efficient transport of assimilates or rice. And talking about the feature cell type A, cell type B, cytoplasm, nucleus, mitochondria, cellulose, and ribosomes. They both got cytoplasms, quite obvious. Nucleus, oh, it does not have a nucleus. It's got cell B has a nucleus. It has many mitochondria. Now, this is very interesting. This means that it needs energy. If it has many, it means that it needs energy. And cellulose, it has it because, well, cell wall, obviously. That's quite logical. Cellulose, all plant cells are made up of, all cell wall of all plant cells are made from cellulose. Okay, this one does not have ribosomes. It has a very few organelles and it does not have a nucleus. Well, if it does not have a nucleus means that the cell is essentially dead. Now it's starting to sound like a xylem vessel, and this probably phloem because here it's called mitochondria for active transport and stuffs. Oh yeah, indeed it is talking about the transport. Okay, so one xylem and the other is phloem. So it says identify the cell A and with type A is xylem. Identify cell type A and explain why plasmodesmata are important. So cell type A is just a xylem vessel. Okay, an explanation why many plasma connect cell type A with cell type B cells. Explain why are they important. Now, I'm just gonna go back to the mark scheme, and these are really notes that you should. These are mark scheme, but you should be making your notes from these wordings really, because this is what you need to be revising. Well, A does not have a nucleus. Isn't that wrong? Did I what's wrong? Something is wrong. A does not have a nucleus. That means that it is dead. Identify cell type A. Okay, A is the mitochondria. How comes that? May plasma desmata connect cell type A cells with type B cells. Okay. Efficient transport of assimilates. Oh, two different types found in plant tissue adapted for efficient transport of assimilates. Okay. Cell type A is dead. Okay. Alright, so apparently it's the phloem, as the mark scheme is saying, but yeah. Cell type A is the phloem sieve tube element, and it connects the cytoplasm of two cells that allow the efficient uh, loading into efficient, you may just say allow the efficient or rapid diffusion of sucrose. Okay, it connects cytoplasm of two cells to allow and hence allow for rapid diffusion of sucrose. Let's just get back here. Sieve tube element. I need that thing. No. Okay, so first of all, is not the xylem vessel apparently. So yeah, the answer is going to be the phloem sieve tube element. All right. And the explanation is that it connects the cytoplasm of two cells. It connects the cytoplasm of two adjacent cells, hence allowing for a rapid diffusion of sucrose, for example. Okay. Well, that's what it wrote there, isn't it? Rapid diffusion of sucrose. All right. Now let us get back to the next question. Next question. All right. Now the liver receives blood from the hepatic artery and blah blah blah. Hepatocytes, main type of liver cell, main cell type of the liver, and synthesis of triglyceride and plasma proteins, detoxifying energy storage. The hepatic artery branches from the main artery that transport blood from the heart. Name the main artery that transport blood to the hepatic artery. It should be the main artery. Well, of course, we have not yet covered that, so I'm just thinking whether I should have included this question or leave it out. Yeah. Now, by the way, where does this come from? Did I not include the answer to that one? What is what year is that? What I put here? It's October, November, QP22. It's about the peroxisome stuff. It's 2023, but the QP22. Yeah, here it is actually. Oh, I didn't put that part. Okay, so just leave it out. Okay. Pardon. Okay. But although the answer would be a branch of the celiac trunk. The celiac trunk. Common. The blood supply to liver is provided both by hepatic and portal vein. Celiac artery. Anyway, leave that now for the moment. Let's focus on what we intend to do really. So now the figure whatever here is a transmission electron. Okay, hepatocyte. Okay, yeah, it's just a continuation of the question above. The peroxisome shown in blah 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 is a spherical organelle bound by a single membrane. It carries out by enzyme catalyzed metabolic reactions, including detoxifications. Indeed, some of these require oxygen. Okay, and okay, so that's a peroxisome. Well, that is interesting because it's got. They mentioned that it needed some sort of oxygen and stuff. So it's got mitochondria here. Mitochondria quite a lot. Okay, now lipid. Describe the evidence visible apart from the presence of peroxisome that indicates some of the functions of a hepatocyte. Describe the evidence visible. So visibly, we can see a lot of mitochondria. So the evidence visible apart from the peroxisome that indicates some of the functions of the hepatocytes. Add labels to identify the location of any if okay, if it is part of my evidence. Okay, yeah. So this would be the mitochondria. You may write just mitochondria. Okay. It's a bit difficult for me to write, but yeah, you write it. And this would be the rough endoplasmic reticulum, by the way, rough endoplasmic reticulum. You write it in full. And what else? I can see the smooth endoplasmic reticulum as well. You can label that smooth endoplasmic reticulum. Again, I'd say you write down the full name, okay, not short form. And either way, it does talk here about the synthesis of triglycerides and plasma proteins and energy storage. Okay, so describe. It says apart from what? It says apart from the presence of peroxisome. So so presence of mitochondria pertaining to the fact that it acts as energy storage, carries that it stores energy. that indicates some of the functions of the hepatocyte. Okay. Presence of smooth endoplasmic reticulum pertaining to its function of lipid. You may use the word lipid or triglyceride synthesis. Okay. We see the presence of rough endoplasmic reticulum pertaining to the fact to its function of plasma protein synthesis. Plasma protein synthesis. Okay. Also quite some points around here. So, anything else we can notice? No, really, not quite, not exactly. Those blackmail structures seem to be, well, can't quite say, but yeah, it's a bit difficult to say. It's called glycogen granules. It says indicates some of the functions of the hepatocyte. So it also, yeah, glycogen granules is what stores that the presence of mitochondria is the fact pertaining to the fact that it you may say it produces that it produces ATP. Presence of smooth. Okay, that's one thing. Then you say presence of glycogen granules. Presence of glycogen granules indicates it pertains to the fact that indicates how it stores energy in the form of glycogen, of insoluble glycogen. Okay, well, that is interesting. Okay, good, there we go. So is it particularly say how much do you need to talk about? It says describe. Okay, all right. So let me see the mark scheme now. All right. Golgi body. Who saw that? No, it was not. It was not. Okay, it says ignore. Exactly. There was no Golgi body, no lysosome, and no vesicle. MP1 and MP2 complete term needed, either in text or labeled. Exactly. Okay, just really make sure to just tie knot with this thing. Always when you're referring to the SCR, the smooth endoplasmic reticulum, or the rough endoplasmic reticulum, make sure to include the complete term. Okay, like smooth endoplasmic reticulum or the rough endoplasmic reticulum. All right, either you labeled it on the diagram or you refer to it in the text. Okay, but actually you are asked to label it on the diagram. Okay, so the smooth endoplasmic reticulum, it involves in the synthesis of lipid or triglyceride. Oh, it does. Oh my god, did I miss the part on the detail? Yeah, I did miss the part on the detail. I forgot to Um, well, anyway, so tubular appearance with no ribosomes, and you may say convoluted stuff and stuff. For rough endoplasmic reticulum, again, it is just flattened sacs, but ribosomes are attached on its surface, and it's here for protein synthesis for ribosome label. Plasma protein synthesis, here you go. For mitochondrion, you form many mitochondrion for to produce ATP, exactly. We say produce ATP, or you say, oh, they are rejecting the term generate energy. They're rejecting the term generate energy. Yeah, you don't make energy, but you can generate energy. Energy can be generated. Generate, like just like we've got generators, they generate energy, isn't it? Anyway, so okay, I'll keep that in mind. So provide energy, or you may say right, aerobic respiration. Okay, so I've included this. I get my mark here, by the way. I forgot the detail. Okay, I used the word protein synthesis. Oh, actually, this one I used. Here I said this, and allow one if and circular. Okay, anyway. A presence of a glycogen granules as is energy store, and yeah, essentially that. Okay, just don't forget to give the details, which I forgot to, but I didn't pay actually I didn't pay attention to that. A part of the question, presence of mitochondria. um you may say rod-like structures for rough endoplasmic reticulum and stuff. Okay, now you've got the idea. You can write it on your own. I'm not going to waste my time on that part. Let us see here. What does the question say? So the mitochondria in the figure are larger than the peroxisome. State one other difference visible between a peroxisome and a mitochondrion. Okay, let's see that. Well, we see the cristae here in the inside of the mitochondrion, but it's not present in here, and I can't quite see, but it's a bit difficult to see. But I'd say about the membrane. Okay, the peroxisome seems to be single membrane, while our mitochondrion seems to be a double membrane structure. Yeah, apart from that, it does seem to be more spherical. It's more of a regular spherical as compared to our mitochondrion. But it says state one other. So one of the most thing that the thing that seems the most relevant. That's what you're gonna put in there as your answer. So I'm gonna say so a peroxisome peroxisome lacks cristae which which on the other hand is present and visible on the mitochondria. Okay, quite clear this part. Absence of cristae or no crista, whatever. So much rolling, eh? Scrolling. Yeah, no cristae. That's it. Double membrane. I did. Yeah, spherical shape indeed. Okay, good. Circular for spherical, sausage shape. Okay, accepted. Okay, cool. What's next? Some of the enzymes used within mitochondria can be synthesized by the organelle. So that is by peroxide. Oh, okay, yeah, by mitochondria itself, indeed, because it's got its own ribosome, so it can remember, enzymes are proteins. Enzymes are proteins. And mitochondria has got its own 70S ribosomes along with its own circular DNA, which allows it to make its own proteins that are enzymes, which it will be using itself. However, peroxisomes cannot synthesize any of the enzymes that they contain. Okay, so if they cannot synthesize, that means that they do not have any ribosomes. Quite logical, isn't it? Yeah. So suggest. Here it says suggest. Why a mitochondria can synthesize, but a peroxide. Okay, quite logical. It's a 2 mark, so make sure to explain sufficiently. Give ample explanation. So this is simply because mitochondria has its own DNA and 70S ribosomes, which ribosomes within it, which allows it to synthesize its own proteins, like enzymes that it needs. However, since the peroxisome cannot synthesize enzymes, this means that they do not have ribosomes within their, you may say, cytoplasm, or just inside their cell. Okay, that's it. Now let us see what does the answer, the key, says about this. So it says mitochondria, or reverse argument for peroxisome, has DNA, code for enzymes, okay, 70S ribosomes. Yeah, obviously you need to reject, you can't use the word ATS, okay, so yeah, any two from, so that's 1 mark here, and that's your second mark here. All right, yeah, that's it. Really, you don't really need much more. So yeah, get the mark here for the moment. Chaperone mediated. Next question. Let us see. So one of the enzymes present in peroxisome is catalase. Oh, that's interesting. This enzyme catalyzes the breakdown of hydrogen peroxide. Yeah, that's the same thing that's present in potatoes, to harmless products. Breakdown of hydrogen H2O. Okay, it suggests why is it useful to the cell for this reaction to take place within peroxisomes. Okay, well, that is very interesting. Well, so let's read it again. So one of the enzymes present in peroxisome is catalase. This enzyme catalyzes the breakdown of H2O2 to harmless products. So H2O2, okay, into harmless products like water and oxygen. So suggest why is it useful for the cell for this reaction to take place within peroxisomes? Well, much simply because it suggests, okay, it is suggest. So first of all, enzymes, they are very sensitive substances, so they do need specific, for example, pH. Okay, they do need specific pH to work. So we could perhaps imply that the peroxisome has the specific, the perfect, okay, the optimum pH to sustain the enzyme catalyzed reaction, simply the enzyme reaction of catalase. So perhaps you may say that peroxisome, peroxisome, they have, they have, peroxisomes have the optimum pH to sustain the enzyme reaction or catalase. Okay, it's called the optimum pH. And what could be another thing? Well, perhaps so that the enzyme does not just sort of leak out, so that the enzyme does not go out into the cytoplasm of the hepatocyte, right? So, and secondly, so that the catalase does not leak out into the cytoplasm of the hepatocyte. Now let us go on and see the marking answers that would give us a very beautiful insight on the type of answers they are expecting. Alright, so the rest of the cell is protected from hydrogen peroxide, okay. Compartment, more efficient, high concentration, okay. Provide optimum conditions for, yeah, I got 1 mark for here. So, and then it says product of oxygen as byproducts are useful for isolate peroxisomes enzyme from the rest of the cell. Yeah, that's what I put, but in an alternative wording, so that that thing, the enzyme catalase, does not leak out into the cell. But these answers are very interesting, like to maintain a high concentration of enzyme in one specific location, so therefore the reaction can occur faster. So more enzyme substrate complex can form, and hence the reaction can occur faster. And this also is very interesting. So the rest of the cell is protected from hydrogen peroxide. So maybe, maybe hydrogen peroxide can have toxic effect on the cell. And well, yeah, essentially that. Now we are reaching the culmination of this paper, with here being our last question. Now let us go and quickly see this last question. It is paper 2023. It is the 2023 paper of the October-November series, and it is the third variant of paper 2. It says new mitochondria are formed when a mitochondrion divides into 2. Okay, that's very interesting, isn't it? So is a blah 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 is a transmission electron micrograph of a mitochondrion that's dividing. Okay, that should be our first, and that's the second mitochondria. Mitochondria 1 and mitochondria 2. It's very interesting. So that's a 50,000 magnification, by the way. Obviously, digitally we won't really be able to do these magnification questions because it'd be difficult to measure, Anyway, it's going to be a bit problematic, but you get the idea, right? How do we do that? So state two features of mitochondria that are visible in the figure. So, well, it's very clear. It's called the cristae and it's called double membrane. That's it. So I'm gonna write so cristae on what did I write? Cristae and then a double membrane. Let me see. Membrane. Okay, now the next question, I mean the last question here of today is that mitochondria, they code for mitochondrial DNA codes for some polypeptides of proteins used within the mitochondrion. Yeah, we saw that recently now about the enzymes. So some of the proteins allow movement of ions. Oh, so it's talking about channel proteins, the ones that allow the passage of some substance. Anyway, outline the ways in which ions can move into the mitochondria. Now, it's not really a chapter that we've already done, but I think we could somehow, get a first initial exposure to that. It's a 3 marks question. So outline the ways in which the ions can move into mitochondria. So we are just going to discuss it from the mark scheme answer itself. Not going to write anything. So here it is. It's a bit things that we've not really covered, isn't it? Oh, by the way, this thing, yeah. Okay, so it's got two membranes or double membrane, you see, and then the cristae. Okay, so it's called the inner membrane is folded, a folded inner membrane. I don't know who saw the ribosomes. Maybe I didn't see properly. Anyway, so what would be the suggestion? Outline the ways into how ions move. So they move by active transport, that is by the usage of ATP against the concentration gradient that we get for mark. Then you may talk about how protein, or you say carrier protein or pump, they just change their shape, binding sites. For channel proteins is more for facilitated diffusion. Okay, that would be my second mark, perhaps. So 1 and 4, and then facilitated diffusion here, then you elaborate a bit here. So no energy and down the concentration gradient will be third mark. Yeah, that's good. Okay, now we are done with quite a few of the questions. Now we have not yet explored, well, till date, what we've done are questions ranging between the year 2023 and 2020. We've worked questions. We've worked questions okay, ranging between 2023. So we've worked questions ranging between 23. So all questions that's based on cells. All questions based on cells from 2023 has been covered, and I think I've gone till 2022. I can't really confirm at the moment if each and every questions of 2022 has been covered in here. I can't really exactly confirm that part, but yeah, there we are. We've completed questions from 2023 to 2022, and we shall be taking a look at all the questions as well. But although I think we've got enough exposure to the contents and the type of question asked, and it is still quite an ample amount of questions that we've practiced together, and it's ample amount of contents as well that you should be including in your notes. So there's a lot to learn from the mark scheme. So make sure to include these stuffs into your notes. Okay, combine these elements into your notes for a more precise knowledge. Okay, so that you don't just revise stuff that you won't really be using. Okay, so revise efficiently. And there we are. Now I haven't really touched paper one questions, but maybe we could take a look at those. Those are a bit more tricky, but um, those are nice as well. Okay, those helps to really get to the really intricacies of the concepts that you've learned. So yeah, it will make you question things that you've never thought about. But it's good, okay, it's good. So anyway, I'll leave you for today, and perhaps what I shall be tackling next is questions from the next chapter, that is biological molecules, especially the part of proteins folding. That's a bit trickier, but we shall be tackling it together. And anyway, for now, I leave you all and keep revising efficiently, okay, smartly. Until then, bye and stay safe and take care and stay healthy. All right? All right. Goodbye. See you soon.